Hello everyone, this is Kasada Owen. Welcome back to the InnoPod, and today I'm here to take you through hemoglobin and porphyrin derived pigments as an extracellular accumulation, basically. So these include the hemoglobin, hemosiderin, hematine, bilirubin, and porphyrins. But if you to realize, all of these ones are endogenous pigments. In this, I mean colored substances formed through the normal metabolic pathways of the body, as you can see, hemoglobin and the others. So many of these pigments are indicative or diagnostic of specific disease entities, in that knowing these pigments and the circumstances in which they occur, they should help one to determine the differential diagnosis and the significance of lesions that are colored. So where you see brown lesions within the cells, where you see red ones, you can realize that this is a disease because of this pigment that has been deposited abnormally in these spaces. So I will start with hemoglobin itself. When it is seen being deposited in between cells, it is seen as reddish brown casts in the renal tubules as a sequel to intravascular hemolysis. So if you realize that when red blood cells are being broken down physiologically in the body, they will produce basically three components, and those are globin, heme, and iron. But this hemoglobin, which has been after breaking down the red blood cells, it will diffuse to the surrounding tissues and give it a diffuse red staining appearance. In most cases, what we call as hemoglobin imbibition. But this usually happens in postmortem changes in a way we can liaise to know about it. So when the hemoglobin further is broken down by lysosomes and other enzymes within the body, it will produce the three components, globin, heme, and iron. So globin for it is a soluble protein, as we know, and in tissue fluids, and is removed from the areas back to blood and lymph. So it goes back to do other functions, still forming more hemoglobin. Then the iron, for it is in total, the total iron is basically divided into two components. We have the functional and the storage iron in the body. So the functional iron is that one that is underused, or it is having a use within the different pigments. So for example, it is found in hemoglobin, myoglobin, and iron-containing enzymes such as catalase and also cytochromes, as we see in the ATP formation. Then the other storage pool for it is represented by ferritin and hemosiderin, as we shall see later on. So we shall start with hemosiderin as another pigment. So all of these ones are hemoglobin derivatives, as we saw. And this one, its color is golden yellow to brown granular or crystalline pigment, in which form iron is stored in cells. So when iron is broken down and it is in excess, it is turned into ferritin ions, which then later on are used to make hemosiderin to store this iron. So hemosiderin is a storage form of iron, as we saw earlier on. But under normal circumstances, it is found in smaller amounts in mononuclear phagocytes of bone marrow, spleen, and the liver, greatest in the spleen of the horse, but least in dogs. Those are species specifications. So excess iron causes hemosiderin to accumulate within cells, but it is basically via two processes. There is the localized process, and also we have the systemic rearrangement. So what is this localized process? It results from hemorrhage. Hemorrhage will mean that the rupture of red blood cells and blood oozes to tissues. And it can also be due to the vesicular congestion, whereby the blood vessels are congested, and as a result they rupture and release these red blood cells, which are going to burst and produce those iron. Or even in some species like the dogs, it can be due to the heartworms, like in the canines. So a common example that we can usually use is the common browse. So the play of color through which this browse passes, maybe when you get a browse on your body, it reflects the changes, these changes. In that originally the browse is red in color. When you have just got it on your skin, it is red, and that represents hemoglobin. But then later on it will turn into green-blue, which shows the local formation of biliverdin, and that is what is called green bile. Then it will turn into red bile, that is bilirubin, and thereafter it will turn into the iron molecule of hemoglobin deposited as golden yellow. Sometimes it can be brown in color, granular substance, which is called hemosiderin. So hemosiderin is observed in scars. So when you have a scar, that color that you see sometimes that is being brownish in color, that shows hemosiderin. Then the other one is the systemic derangement, which results from the excessive consumption of iron and also due to the impaired utilization of iron, but it can also be due to the hemolytic anemia and transfusion. So here, hemosiderin is deposited in many organs and tissues, a condition called hemosiderosis. That is in but the extreme iron overload is called hemochromatosis and is associated usually with the liver fibrosis, that is its swelling, and then diabetes mellitus. Yeah, basically these ones shall be explained later on in the liver pathology as you follow us. So what is the microscopic appearance? You will be seeing coarse golden, yellow, or sometimes brown granular pigments lying within the cytoplasm of cells. But this, so hemosiderosis is diagnosed in the tissues using the Prussian blue reaction due to the ferric ions that are within this hemosiderin, so which is stained positively with this Prussian blue staining. So this is what we shall be seeing in the microscopic appearance. You can see these brown depositions within cells here and here and here. That is hemosiderin, basically even here. So this is a kidney, as you can see it, because of these renal corpuscles. They are here being deposited. Yes. So then you go to another one, which is hematin, formalin pigment, and the parasite pigment. So hematin is just a non-specific group of brown hemoglobin derived pigments which stain negatively for iron. This is because the iron that is within hematin is tightly bound and therefore it is unreactive with the Prussian blue stain as we saw in hemosiderosis. So then we have another pigment which is called formalin pigment or acid hematin. So this one occurs in blood-filled spaces, tissues, fixed in unbuffered acid formalin. So granules are extracellular and can make histopathology interpretation very difficult. Yeah, because of these granules being extracellular. So we have then the next is the parasite pigments. So these can be many depending on the parasite that is infecting the tissues or invading these tissues, like the Plasmodium, the Fluke fasciolides magna, the human blood flukes, and many others. When they come into these tissues, they will deposit certain pigments, and these will show colorations within changing colors of these of these tissues. So then the next we have bilirubin. So bilirubin, this one is also a derivative of hemoglobin, but it contains no iron. So when there is bile obstruction, it leads to retention of bile, a condition known as cholestasis, that is in the bile canaliculi and the ducts in the liver. Then if there is accumulation of bilirubin in tissues, it gives the yellow color to the skin, the sclera, mucous membranes, and this is a characteristic. These are all characteristics of jaundice, or what we call iterus. So, and there are basically three types of iterus or jaundice. We have the prehepatic, which is known as the hemolytic jaundice, and this is due to the overproduction of it. Can be due to these basic reasons. So basically, they all result from the hemolysis of red blood cells. But this follows three mechanisms. It can be due to the overproduction of bilirubin, due to the lysis of red blood cells, or reduced hepatic uptake of bilirubin, and also it can be due to the impaired conjugation of bilirubin. Then the hepatic one, which is also called the hepatotoxic jaundice. So as you hear the name toxic, this results primarily from the injury of the hepatocytes with now silage of conjugated bilirubin into blood. So when the liver cells are damaged, this bilirubin will be released to other tissues and it will go on staining them. And here, both unconjugated and conjugated bilirubin will accumulate in blood and also other surrounding tissues. Yes, in most cases we call this bioinhibition sometimes because tissues take up this thing. Then in the next one is the post-hepatic or obstructive jaundice, and this one results from a reflux of bile pigments into blood when the bile ducts are obstructed. So if there is obstruction of the bile ducts, the bilirubin will be now taken up by other tissues that will diffuse out. And this is a characteristic of conjugated hyperbilirubinemia. Yes. Then the next pigment is porphyrins. These ones we say that they are tetrapyral ring structure. Sorry, that word is ring. Yes, related structurally to heme of hemoglobin. So they resemble heme. The way you can see the structure of heme is the same way they resemble. So now the condition in which there is accumulation of these porphyrin pigments is called porphyria. Yes, and then the most common one is the congenital porphyria, which is a metabolic defect in synthesis of heme and results in accumulation of heme precursors in blood and tissues. So if there is a defect in the synthesis of hemoglobin, or I mean heme, then you see that the precursors will start accumulating, and these are basically the porphyrins, where we have urobilinogen and many others. So then if these accumulate, then the accumulation in the bone and teeth gives these structures dark pink color, and that's the name pink teeth. I didn't include the picture, but when this is mostly observed within the herbivores. Then we have porphyrin derived pigment. What you were seeing earlier on were the hemoglobin derived pigments. Now here they are the porphyrin derived pigments. So one most common one is the pyrroerythrin, which is a plant pigment that arises when chlorophyll is metabolized by the ruminomicrobes, and then it is transported to the liver for degradation and excretion. But in most cases, if there is hepatotoxic hepatocellular injury, this pyrroerythrin is going to accumulate within tissues. And when it accumulates within tissues, it will react with the sunlight in a process known as pharmacodynamics, resulting into severe injury of cells in the skin. And as a result, this is going to cause photosensitization. Photosensitization, whereby the tissues can no longer sustain the ultra radiations that come from the sun. So the result will be that these animals will be suffering injuries as the skin sloughs off. Yes, and they get wounds on the body. So, and the best plant that has most of these hepatotoxic pigments is the Lantana camara. So it is the major one that can cause photosensitization, as you see in the picture here. This is a horse, and majorly it affects those spaces which are white in color because they have little or no melanin. So that's why it is easier for them. Now, areas where there is melanin, like these which are dark, it is hard for there to get wounds, but these areas where it is white. So this majorly affects those species of animals that are exotic. Yeah, it is more of those animals. And even here you can see the skin sloughing off, the cow remains with wounds generally. Yeah, so that is what I had for you about the endogenous pigments as uh, accumulations in the body and tissue. Thank you so much for listening. Hope to see you in the next presentation as we share. Join and subscribe the Innovates. God bless you.